Welcome friends. I am Leema Rosemary, Assistant Professor, Department of Biochemistry, St.Joseph College of Arts and Science, Cuddalore. Now I am going to talk about heme catabolism. The lifespan of erythrocytes have 120 days. At the end of this period, they are removed from the circulation. Erythrocytes are taken up and degraded by the macrophages of the reticuloendothelial system in spleen and liver. The hemoglobin is cleaved to the protein part and non-protein part. Protein part is a globin and non-protein is heme. About 6 gram of hemoglobin per day is broken down and resynthesized in an adult man. 80% of the heme that is subjected for degradation comes from the erythrocytes and the rest of 20% comes from immature RBC, myoglobin and cytochromes. This is the structure of hemoglobin, and that is heme with globin. Globin is a protein part, heme is a non-protein part. Heme contains 4 pherol ring connected by a methyl bridges and centrally contains Fe2+. Role of heme oxygenase. A complex microsomal enzyme, namely heme oxygenase. It utilizes NADPH and oxygen and cleaves the methyl bridges between the two pherol rings, that is A and B, to form biliverdin. And biliverdin is a green pigment. Simultaneously, ferrous ion is oxidized to ferric form and released. Heme promote the activity of this enzyme. Biliverdin is excreted in birds and amphibians, while in mammals it is further degraded. This is overall heme catabolism. Biliverdin reductase. Biliverdin converted to bilirubin by biliverdin reductase. Here, methyl bridges between the pherol rings C and D are reduced to methylene group to form bilirubin. And this pigment is yellow color. The enzyme biliverdin reductase is an NADPH dependent soluble enzyme. 1 gram of hemoglobin on degradation yield about 35 mg bilirubin. Approximately 250 to 350 mg of bilirubin is daily produced in human adult. The term bile pigment is used to collectively represent bilirubin and its derivatives. Transport of bilirubin to liver. Bilirubin is lipophilic and therefore insoluble in aqueous solution. But bilirubin is transported in the plasma in a bound to form albumin. Albumin has two binding sites for bilirubin, that is a high affinity site and low affinity site. Approximately 25 mg of bilirubin can bind tightly to albumin per 100 ml of plasma. The rest of the bilirubin bind loosely, which can be easily detached from albumin to enter the tissues. Certain drugs and antibiotics can displace bilirubin from albumin, that is sulfonamides and salicylate drugs. Due to this, bilirubin can enter the central nervous system and cause damage to neurons sometimes. As the albumin bilirubin complex enters the liver, bilirubin dissociates and is taken up by sinusoidal surface of the hepatocytes by a carrier mediated active transport. The transport system has a very high capacity and therefore is not a limitation for further metabolism of bilirubin. Inside the hepatocytes, bilirubin binds to a specific intracellular protein, namely ligandin. Then conjugation of bilirubin. In the liver, bilirubin is conjugated with 2 molecules of glucuronate supplied by UDP glucuronate. This reaction is catalyzed by bilirubin glucuronyl transferase results in the formation of a water-soluble bilirubin diglucuronide. When bilirubin is excess, bilirubin monoglucuronide also accumulate in the body. The enzyme bilirubin glucuronyl transferase can be induced by a number of drugs, example phenobarbital. Then excretion of bilirubin into bile. The conjugated bilirubin is excreted into the bile canaliculi against a concentration gradient which then enters the bile. The transport of bilirubin diglucuronide is an active, energy dependent and rate limiting process. This step is easily susceptible to any impairment in liver function. Normally, there is a good coordination between the bilirubin conjugation and its excretion into the bile. Thus, almost all the bilirubin that enters bile is in the conjugated form. Fate of bilirubin. Bilirubin glucuronides are hydrolyzed in the intestine by specific bacterial enzyme, namely beta glucuronidase, to liberate bilirubin. The latter is then converted to urobilinogen, a small part of which may be reabsorbed into the circulation. Urobilinogen can be converted to urobilin in the kidney and excreted. The characteristic of color of urine is due to the urobilin. A major part of urobilinogen is converted by bacteria to streptobilin, which is excreted along with fecus. The characteristic brown color of fecus is due to streptobilin. This is the pathway of heme catabolism. Initially, matured RBC degraded in macrophages. Here, hemoglobin degraded into heme plus globin. Then heme cleaved by heme oxygenase to form biliverdin. Then biliverdin converted to bilirubin by biliverdin reductase, and the bilirubin is a insoluble form, but then it combined with albumin complex in blood, finally transferred to the liver. Here bilirubin conjugated with 2-UDP glucuronide to form conjugated bilirubin like bilirubin diglucuronide. Then the bilirubin, actually bilirubin is the waste excretory product, so it reaches the intestine and kidney. Here the microbial enzyme, it converted to urobilinogen, then urobilinogen converted to urobilin in the form of the excreted form in urine, and microbial enzyme, it converted urobilinogen to streptobilin. This streptobilin excreted through fecus. These two pigment to give color to the urine and fecus. Thank you.